Welcome to Going Antiviral, the podcast for the IAS-USA, a professional continuing medical education organization focused on HIV and other viral diseases. I'm Dr. Michael Saag, professor of medicine and infectious diseases at the University of Alabama at Birmingham and volunteer member of the IAS-USA Board of Directors. Welcome to the Going Antiviral podcast. Today, March 31st, 2025, we are speaking to Dr. Graham Hatfull, a professor of biological sciences at the University of Pittsburgh. Dr. Hatfull focuses on the molecular genetics of the mycobacteria and their bacteriophages and their use for educational advancement and clinical intervention. Today, Dr. Hatfull joins us to discuss bacteriophages and the impact of antimicrobial-resistant bacterial infections on global morbidity and mortality. Graham, welcome to the podcast.
Hi, Michael. Delighted to be here.
It's wonderful to have you. So let's start with the basics. What are bacteriophages in a phrase or two?
Well, bacteriophages, or commonly just called phages, are viruses, and they share all of the properties that we understand viruses have, but they infect bacteria. So they're viruses that infect bacteria.
Right. And how did you get involved in working with phages?
Well, we've been working with phages for a rather long time now. We got interested in all of their aspects, their genomes, their diversity, how they've evolved, how many different types there are, how they go about infecting cells. And so we've very much been interested in pretty much every basic question that you could imagine. And our entry point was we were trying to study a particular recombination system, but we quickly fell in love with every single aspect of phages.
That's great. And shows what academic freedom and just kind of following your curiosity and where it leads you.
You bet. Absolutely.
Yeah. I mean, I first started working with phage when I was in a molecular biology lab and we were trying to clone certain aspects of gene products, and we'd go in and look for plaque forming units on the next morning after we set up cultures, and it was always disappointed when we didn't have plaques. That was the bane of my existence for a while.
Well, we've isolated lots of phages by essentially doing exactly that. So it really does work.
Exactly. So let's talk a little bit about how phages can be used clinically, and in this case specifically for antimicrobial resistant bacteria.
Yeah, so the idea of using phages therapeutically is a very old idea. It was really pioneered by Felix Durrell and colleagues way back around the 1920s, sort of shortly after phages had been discovered. And so, yes, you may ask, well, if people have been thinking about them as therapies for 100 and something years, why aren't we using them? And that would be a very good question. It's been a long and rather complicated history where, although they were considered as antimicrobials, antibiotics came along, and the Western world took a good advantage of them, while in the parts of the former Soviet Union, for example, they continued to use phages therapeutically for some time. More recently, there's been renewed interest because of the concerns about the ongoing march of antibiotic resistance amongst bacterial pathogens, and the need to figure out where the next types of therapies are going to come from. And although the efficacy and the utility of bacteriophages is, I think, still somewhat unclear, there's considerable hope that there could be types of infections and types of pathogens for which phages could find a role as therapists in treating those infections.
And as a rule of thumb, when a phage binds and enters a bacterial cell, it usually causes a lytic phenomena. It blows the cell up in some sort of way.
Yeah, we would think of phages mostly being grouped into two sort of major groups. There's the so-called lytic phages, who do exactly what you described. They inject their DNA typically. the DNA replicates. They make more copies of themselves, and then when they've made perhaps 100 new phage particles, that cell lyses, meaning it destroys itself. The bacterial cell dies, and the progeny phages are released. But there's a second class of phages that are called temperate phages that are very common. They can do that process, but in a rather complicated form of their biology. They can also form what are called lysogens, sort of a parasitic state where the phage just goes along for the ride without killing the bacteria, and just replicating along with it. So temperate phages are not the kind that would typically be used therapeutically, except with modern engineering tools and an understanding of the biology. You can essentially fix them so that they stop that lysogeny business, and then they behave just like a normal obligatorily lytic phage would, and therefore could be used therapeutically in principle.
Right. So I guess the part that, at least when I first started thinking of this as a therapeutic, I thought, okay, so you just find some bacteriophage or some phage, you infuse it, and boom, whatever's evil and bad it's gone, and you move on. But it's a lot more complicated, right? It has to be like a match between the phage and the specific bacteria, correct?
Yes, so this is the classic double-edged sword. The wonderful thing about bacteriophages, and a big advantage over antibiotics, is that they're very specific for the bacteria that they infect. So when you take antibiotics, they tend to kill all sorts of bacteria in your body. Your microbiome in your gut gets wildly distorted and upset, and so you have lots of common side effects. Phages should be, and typically are, very specific. So you should be able to use them to go in as a targeted missile and take out specifically just the bad folks.
So it works kind of like a smart bomb. And
Yeah, just like a smart bomb. The problem is that the specificity that gives you those advantages is also a problem, because they're often too specific. That means that they may only attack one or a particular type of strain of a bacterium, but not a different strain. So if you and I both had a particular type of bacterial infection, there may be phages that would be useful to try to treat my infection, but they wouldn't be useful for yours because they simply don't infect those. And so
so
that's a real it's a problem that's arisen in the implementation.
So let's carry that forward a little bit. Let's be specific in this case you gave. So we both are infected with an Acinetobacter, say, and that Acinetobacter in your case and in my case seems to be heavily resistant to common antimicrobials. So we make a phage that actually works for your Acinetobacter, but for mine it might not work, correct?
Yeah, that's exactly right. And I could just drill down to a very specific case, because my lab works on pathogens in the genus of the mycobacteria,
Yes.
bacteria that cause tuberculosis and a number of other infections, um, including those that are quite common with cystic fibrosis patients. And I the specific example I would give is back in late 2017, we were sent two bacterial samples, two isolates from two patients who suffered in a very similar way. Both had cystic fibrosis, both had a bilateral lung transplant, but then had these disseminated infections. And so we went looking for phages. We were lucky to find phages, and we engineered some phages that would work for one patient, and they seemed to work quite well for that one patient. And we never did find um, uh, phages for that second patient, and she unfortunately passed away a couple of months after we had been looking. So it just helps to illustrate both the severity of these types of diseases when you can't control them with antibiotics, and the fact that this matching is often a matter of life and death in terms of the outcomes of whether you can find a match or not.
So is it a matter of trial and error, or do you look at receptors and try to gauge based on the structure of a receptor, that one's likely to accept this type of phage or that?
That's a wonderful, wonderful question. So, what we and others do quite routinely is to test in the lab specifically whether a set of phages that we have in our collection will or will not infect a particular clinical isolate from a strain. Of course, what you'd like to be able to do, and there's a lot of quite high level of interest in this, is to be able to just use information that you have about the bacterial strain, maybe if you have a genome sequence, to be able to predict, perhaps using machine learning, artificial intelligence type strategies to say, aha, I can see the sequence of this bacterium. We got that quite quickly. And now we can use that to predict that phage X, Y, and Z in the library will work really well, and then essentially go straight into a patient with that. That turns out to be, it's an interesting question. We're looking for, we and others, the community, are looking for answers to that. One of the complications is we know that whether a phage infects a strain does depend on the receptor or whatever molecules on the surface the phage binds to, but it also depends on a whole plethora of sort of defense mechanisms that bacteria use to defend against phage infection, things like CRISPR. Many people have heard of CRISPR because it's used for engineering tools, but bacteria naturally carry CRISPR systems which protect from phage infection. And so the real question is, when will we have the genomic power to make accurate predictions, given that there are so many different determinants of whether a phage infects a bacterial strain or not? This will probably be easier for some types of pathogens than other types of pathogens. I'm hoping that for some of the easier ones, we'll get there relatively soon, and others might take a while.
Right. We've had an episode or so on CRISPR-Cas9 and using it for purposes of gene therapy, hypothetically, if not in reality. So you're saying that the natural production of CRISPR actually occurs in bacteria in this case, and one bacteria might just happen to have a CRISPR that cuts up the genetic material of the phage, and then therefore it doesn't work.
Yep, exactly. And that's just one example of many different types of defense systems. And so understanding that whole sort of panoply of really defense systems and the whole dynamics and which molecules and genes determine whether a phage infects one strain or not a different strain. We need to have a much better understanding of all of that part of the biology.
And I'm just thinking further on this. So it may not even be in the bacterial genome itself, right? It could be a plasmid or something that is inside that bacteria that one strain of, in our case, Acinetobacter might have that and
Yep.
the other one not. And that explains why phage one might work in one situation but not in the other.
So absolutely, but this gets marvelously complex as well, right? So it could be that, and we know that phages carry systems to counter the defense, things like anti-CRISPR, right, or anti-restriction modification, and so that's what 3 billion years of evolution at 10 to the power of 23 infections per second will give you this narrow specificity and this massive complexity of the interactions. What we're looking at is the biology of this constant warfare between the bacteria and the viruses, and which one is going to win.
Yeah.
It's super interesting from a basic biology point of view, but from a therapeutic and a translational point of view, we gotta get a better understanding of all of that stuff.
And despite all the political rhetoric about diversity, there's diversity in nature, whether we like it or not.
Yeah, phages are just wonderfully diverse in terms of their genomes, and so I just say briefly, we've been involved in the isolation and characterization of a fairly large number of phages because of the education programs that we've helped to contribute to, and so the thousands of students out there have isolated, and we've sequenced something like 2,500 individual phages that infect a particular strain of mycobacteria. And of those 2,500, I think we only have 2 instances of exactly the same phage being isolated at different times by different students. And so the diversity encompasses, being different by a few SNPs, or being different by having no genes in common whatsoever. Just a massive range of diversity, enormously interesting. And of course, it tells us something about how we think they're all evolving as well.
Let's dig a little bit more into the phage itself. So, we've all learned a lot about bacteria. I mentioned there are certain genes that can jump from bacteria to bacteria. Does that same phenomena happen in phages? Seems like they're kind of smaller, not quite as amenable to interference or some sort of extra genetic material coming to them. Is that correct?
Yeah, so what we know from looking at phage genomes and comparing them is that they appear to be built, so their architecture are as mosaics. So we think of mosaic art from the Romans, for example, where different colored stones and tiles are put together in order to make a picture, right? But those same stones and tiles can be rearranged to make different types of pictures, depending on how you arrange them. And phage genomes are just like that. They're genes which are being swapped around in the population and put together in new combinations. And so that exchange of genes between phages is what has driven the evolution and has been a dominant force in how phages have evolved. So it's not just nucleotide changes from generation to generation; it's whole pieces which are being swapped in and out of different types of genomes. Just giving them this marvelous diversity and this opportunity to sample broadly, to get the genes that they need that make them successful.
And with the degree of replication that likely goes on inside of a, let's say we give this as a therapeutic to a person, certain set of phages, probably homogeneous, you infuse that, it starts infecting the bacteria. But as you said, in the lytic phase, it builds up and then explodes out and has lots of progeny.
Yep.
These progeny are likely to be like the parental phage, but I suspect some diversities sort of creeping along as it's replicating at high levels inside that first bacteria that it infects. So, can you get genetic drift inside of a single patient who's had a phage replicating over time?
So that's a really interesting question, and in many ways, you would expect that you would see at least some drift or changes actually due to selection under those particular circumstances. Um, in reality, in many therapeutic cases that have been reported, um, it's not always easy to recover large amounts of phage particles in order to do that analysis,
Analysis.
and that's partly because whereas you're absolutely right that one hopes that you get some local amplification of the phages at the site of infection in the body. The phages don't just sit around; they get absorbed and they get taken up by different parts of the body, and um, it sort of emphasizes another facet as we think about using phages therapeutically is that understanding the pharmacokinetics, where the phages go, what the concentrations are, how long for, um, is quite complicated. It's a very different ball of wax than looking at pharmacokinetics of antibiotics, for example.
Got it. Shifting gears a little bit, now let's go back to the clinical setting. Let's say you're working as a resource lab for a um, development of specific phage therapy for an individual patient somewhere in the United States. They send you the sample of bacteria that's resistant. Walk me through the process of what you would do in the lab to start finding that right phage for that specific bacteria.
Yeah, so we do this quite a lot in my lab, and over the last 5 years or so, have received something like 500 isolates of Mycobacterium. And so what we do is we grow the bacteria. We'll grow it on a petri dish so we can look at the bacterial colonies. That can be useful to understand about what types of bacteria we have. We grow them in a liquid culture, And we do a series of analyses where, first of all, we'll put the bacteria onto a petri dish to form a lawn. So just a sort of a solid lawn of bacteria growing there. And then we'll spot different phages on there, and we'll ask which ones essentially form a spot or a plaque, as it's called, which would indicate that the phage is infecting and replicating. So typically what we do is we have a set of 3 dozen, it may be almost 4 dozen, which will be our favorite candidates for phages that might work. I mean, we have 28,000 phages, but most of them are not going to work, and we've whittled it down to a set. It's constantly growing, and we're constantly looking for new types of phages and engineering them. But that's what we'll test first of all. For some strains, we won't get any hits at all. For other strains, we'll get one or more hits where it's clear that a phage infects that strain efficiently. That's not good in our minds. That's not good enough, though. We want to know not only that the phage infects, but we also want to know that it actually kills the bacteria, and it kills the bacteria without a lot of resistance to the phage emerging. And so we do a set of experiments where we take the bacteria, we add the phage that looks like it works, and we just try to kill as many of the bacteria as possible. And then we have a look and ask who has survived and at what frequency. And so we try to whittle it down to a particular isolate for which we have one or more phages, which infects nicely, but also efficiently kills those cells in the lab, and does so without any substantial level of resistance occurring to the phage. Those are the good ones. Those are the ones that we want to then have a conversation about what a therapy might actually look like.
And then you found that super hit, that one, so let's say almost perfect phage. Then you amplify it and create it, and you have to have a certain you measure it as colony forming units, or wouldn't be that, but plaque forming units.
Yeah.
That gives you a relative concentration that you would think would be required for infusion into that patient.
Yeah. So we amplify them and we purify them, as you said. And the nice thing about growing phages is that you can grow them to quite high concentrations. And so we can make preparations that have up to 10 to the power of 14 particles per mil. And we typically prepare those out into glass vials in which we lyophilize the phage particles. So we dry them with some excipients, some chemicals that are safe to make sure that they are stable. And typically each one of those vials would have about 10 to the power of 10 particles per mil. And that's enough for a standard therapy for about 10 doses. Okay, so that's how we do that calculation. And most of the patients that we've provided phages for are receiving the phages intravenously. They don't have to be, but for most of these infections, that's what we do at a dose of 10 to the 9 PFU per mil twice a day. So that vial has enough for 5 days worth of administration. For these types of infections, patients are usually taking the phages for 6 months or more. So it's a fairly serious endeavor, but I think at least with some success.
Huh. So I was thinking, though, is if you give these phages at that dose, that once it starts infecting the bacteria successfully and it goes through a lytic phase, it would be generating more phages, so you might not have to give them as often or maybe ever again.
Now that's right. And
That's wishful thinking.
the question is, how are we going to find out if that's true or not, right? And so what you'd like to do, let me just backtrack and just say that when we say we use 10 to the 9 plaque forming units per dose, and we do it twice daily, those are both just rank guesses because we don't know what better to do. That's a manageable regimen, but we don't know that it's any better than one dose of the same amount for one day, or whether it would be better if we gave 10 times as much 5 times a day.
Yeah.
We just don't know. And so what's needed is to get to a position where you can do clinical trials, where you can do them in a well-defined patient cohort, and and just use the clinical trials to stepwise change the variables and understand them. Right, we can do exactly what you suggested. You could compare a regimen of just a relatively high dose once or twice versus a standard regimen over a longer period of time, for example. And once we get to that point, we'll begin to actually have data that we need, some real science to tell us what is the most effective.
So we're really at the beginning of this frontier, even though 100 years ago the idea was thought of. It's really now just beginning, coming into its infancy. It sounds like.
Yeah, well, so I think that's absolutely right. One of the things that this therapeutic use of the phages has going for it is that the collective experience is that, provided that they're purified appropriately, the phages appear to be very safe. We're always worried about safety, right? First, do no harm, and we worry about safety, but our growing experience, I mean, us as the community, is that they are generally very safe. And that means that there's an opportunity to at least use them, as we have tried to contribute to, for compassionate use cases for those patients who really are completely out of choices because the antibiotics have failed. And because the phages can be given safely, it's an opportunity to have a look and see um, if you have any beneficial outcomes, and to try to learn from doing that, so that you can then use that information to design good clinical trials. But we're very much at the front end of what could be done, and we're hoping that this will be a period of time when we'll see a lot of further development and to get some answers.
It's a really exciting frontier, but I'm just flashing back to your description of what you do as a clinical lab to create this. That sounds like it takes a good bit of time. You've picked probably one of the more difficult bacteria with mycobacterium because their doubling time is kind of slow.
Yeah,
Forget about MTB, but even Mycobacterium avium complex can take a couple days to grow out, right?
yeah, yeah,
And so if you had MTB, it could take quite a while for
yeah.
you to develop to do that process, right?
Yeah, so we're very interested in the use of phages for tuberculosis itself. Fortunately, this variation amongst clinical isolates, which is really sort of profound for Mycobacterium avium, Mycobacterium abscessus, appears not to be the case for TB. In other words, we think that TB is a case where a rather small cocktail of, 3 or 4 or 5 phages
Oh,
would actually be effective for most TB patients.
wow.
And to and so that sort of presents itself as an opportunity to do clinical trials, and we're working with colleagues where we're driving towards being able to do that, hopefully sooner rather than later. Um, Oddly, TB might be a really good test case to see, and to understand more about how phage therapy might be effective.
That's really exciting. Our time has flown by as we thought it would. And I think you've kind of already given a bit of a glimpse into the future, but I'm just, maybe you can expand a little bit more on, you mentioned AI and maybe some sort of machine learning or some approach that will speed this up. How would you see that playing out in your lab as you start to assess the phages and the bacteria?
Yes, so I think in general, obviously understanding this question of the determinants of specificity is important. Um, but I think that, in terms of as a medical intervention, using these things that we dig up out of the ground, and most of which we don't understand, and 70% of the genes we don't know what they do, doesn't seem to be a very, doesn't sound or feel like a 21st century technology to me. I think that what we need is to use this experience to move towards building well designed, um, phage like nanomachines, which do all the things that we want them to do well, carry none of the genes that we don't know what they do, and to build them and design them with payloads that facilitates the broadest host range possible. So building synthetic phages by design, optimizing their power, optimizing their host range, optimizing their ability to kill the bacteria. I think that's all well within range. And I think that once we sort of combine the technology for doing that with our understanding of what the genetic determinants are, put those two things together, I think that's where we're going to find useful, broadly used new antimicrobials based on phage biology.
Graham, I can't thank you enough for being with us. It is not only a frontier, it's an exciting frontier. Everyone listening can imagine uses for this, but the angel's in the details, right? Getting it just right and learning as we go. It feels like you're in an exponential growth of knowledge about this, and we all wish you well as you go, and we're happy to go along for the ride as you guys lead the way.
Oh, lovely talking to you, Michael. Love talking about phages.
Well, we loved hearing about it. Thanks so much, Graham.
Thanks.
Thank you for listening to this episode of Going Antiviral. Catch up on earlier episodes wherever you listen to podcasts, and check out the video versions of this episode and others on the IAS-USA YouTube channel. You can find these links in the show notes, or simply go to YouTube and search for IAS-USA, and there they are. Be sure to subscribe, rate, and review Going Antiviral on Apple Podcasts. This podcast is for informational and educational purposes only and is not intended to serve as medical advice. Opinions expressed in this podcast do not necessarily reflect the opinions of the IAS-USA or its affiliates. If you have a question you'd like answered on the show, you can send it to podcast@iasusa.org to be answered in a future episode.